Hello, everyone. Thank you for attending this presentation. Today, I will be talking about the use of solid phase microextraction as a sample preparation technique to overcome matrix effects associated with multi-residue analysis of veterinary drugs in animal tissue. A quick outline of what I'll be talking about today. First, I'll be talking about targeted analysis of veterinary drugs using SPME coupled to liquid chromatography and mass spectrometry. And next, I'll be talking about direct coupling of SPME to mass spectrometry. I'm going to be talking namely about two techniques, coated blade spray, CBS, and direct analysis in real time, or DART. And finally, I'll be talking briefly about untargeted metabolomics in order to detect meat adulteration. First, I'll start with the automated high throughput SPME. This is here what a high throughput SPME brush looks like, where we have here a brush that consists of 96 SPME rods. Each SPME pin or rod is made of a stainless steel that has been coated with an extractive phase. In our case, for this study, multi-residue veterinary drugs. In order to cover a wide variety of polarities, we used hydrophilic, lipophilic balance particles that have been prepared in-house. The length of the extraction phase is 20 millimeters and the thickness is 60 micrometer. This is what a pin looks like, and this is what the coating surface looks like under the microscope. In order to prepare the meat samples for extraction by SPME, first we homogenize the meat samples either in a food processor with dry ice or in a cryogenic mill. After homogenization, in order to prepare the matrix matched calibration samples, we weigh 2 grams of the homogenized meat, spike it with our desired concentration of target of analytes of interest and our internal standards. For this study, I used only one internal standard. After spiking, we vortex the sample for 1 minute, place it on a benchtop agitator for 1 hour, and then let it equilibrate overnight in the fridge. The reason for this, leaving it overnight, in order to achieve equilibria between the analytes and the matrix, in order to imitate contaminated samples or the real incurred samples. The next day before extraction by SPME, we remove the samples from the fridge, bring it to room temperature, dilute it in a 1:3 ratio with water, vortex the sample in order to obtain a homogeneous slurry where we can transfer by pipette 1.5 milliliter of this sample into 96 well plates, and then we place the 96 well plate in the orbital agitators of the Concept 96, and we run the cycle program. What is the Concept 96? Here is a picture of the automated Concept 96 auto sampler. As we can see, it's made of 4 stages. The first one is conditioning, where the extractive phase of the SPME is conditioned in a mixture of methanol and water, typically for 30 minutes, followed by extraction by exposing our extractive phase to the samples that we prepared in the previous slide. After extraction, we quickly rinse the extraction phase for 10 seconds in water, just in case there was a mechanical attachment of our sample to the surface of the coating, where 10 seconds is enough to dislodge any mechanical attachment if it happens. After this quick rinse, we dissolve desorb our analytes in our desorption phase, which is for this specific study is made of acetonitrile, water, and methanol. And after the 20 minutes desorption, we place the whole 96 well plate in the autosampler of the LCMS for further injection and analysis. This protocol here that was developed for both chicken and beef matrix has been studied and compared to two other methods that are considered the golden standard in multi-residue analysis. Namely, these methods are catchers and solvent extraction. The results of this study or comparison have been published, as you can see in the papers that are mentioned here at the bottom of the screen. The first, to summarize the results here, as we can see, the matrix effects in both beef and chicken tissue. In both cases, SPME showed how minimal the matrix effects when using SPME compared to the other two techniques, where the majority of the analytes fell within plus or minus 20% matrix effects compared to catchers and solvent extraction, where a lot of analytes fell outside these ranges. And of course, as expected, when we are able to control matrix effects, then automatically the figures of merit for the method automatically is improved. And as we can see here by comparing the linearity, where we see here in this study, we studied 102 analytes. And we see here in case of beef tissue, 99 out of the 102 analytes achieved a determination coefficient R squared, which is better than 0.99 compared to 87 analytes using catchers and 80 using solvent extraction. And similar results were also achieved with chicken tissue. In terms of accuracy and precision, we can see here the top two figures is for accuracy and precision in beef and chicken, that the samples that are spiked at 0.75, the MRL value or the tolerances, and the bottom two is for samples spiked at 1.5 times the MRL value or the tolerances. And as we can see again, when comparing SPME with the other two techniques, the majority of the analytes fell within the quantitative range, which is 70 to 120% for accuracy and less than 25% RSD. And we can see that in all cases, at all concentrations, and in both matrices, SPME performed much better than the other two techniques. Next, I'll be talking about direct coupling of SPME to mass spec. The first technique I'm going to talk about is coated blade spray. For those of you who are not familiar about the concept of coated blade spray, coated blade spray is basically an SPME-based technique, but in this case, the substrate or the stainless substrate is in a shape of a sword or a blade, where part of this blade is coated with the extraction phase. And of course, the choice of extraction phase depends on the analyte of interest. And once we expose this extraction phase to our sample, then the analytes migrate to the coating, and then these analytes are further desorbed directly in front of the mass spec using the optimized desorption solvent, which is typically a very small amount, 10 to 20 microliters. For this specific study for multi-residue analysis in beef tissue, due to the nature or the complexity of the beef matrix and animal tissue in general, and due to the sharp edges of the blade, the risk of attachment of the matrix to substrate is higher. So as a result, we have optimized the design as we see in this slide here. We optimized the design of the CBS. So instead of directly applying the coating to the blade, we first apply a biocompatible non-extractive phase, such as polyacrylonitrile, and then on top of that, we apply our extractive phase. And in this case, again, for multi-residue, it is hydrophilic, lipophilic extractive phase in order to achieve extraction for a wide range of analytes, a wide range of polarities. Why does this design why is it useful for multi-residue and animal tissue? As we can see here, this is the position of the blade in the sample. As we can see here, the bottom part, which is the extractive phase, is fully immersed in our sample and in contact with our analytes. However, the part of the blade that is still in contact with the matrix is now protected with a biocompatible layer, a smooth layer. So this minimizes the attachment of our matrix to the stainless steel directly, because the stainless steel is now deactivated. And even if there is a mechanical attachment, then a quick rinse will quickly dislodge any mechanical attachment to this blade. The workflow for CBS is similar to what I discussed before with the LC method, but here we have only 3 stages because the desorption takes place in front of the mass spec. As you can see here, first we condition the blades, then we expose them to our matrix, and then we quickly rinse with a solvent. And in the case of coated blade spray, we used 15% acetone in water rather than just water. And after this quick 10-second rinse, the blades are desorbed directly in front of the mass spec. The main benefits of this technique is that we can apply both negative and positive modes right after each other. We can start with negative and then positive, or positive and then negative. We tried both and both works. But the main advantage is that we can use the same blade, the same device, the same sample, same desorption solvent in both modes, and both modes take less than 1 minute analysis, as we can see here. In terms of figures of merits, first of all, we looked at the matrix effects. Although the matrix effects, of course, is not as good as the LC technique, and it's expected because we don't have chromatography here, but still we think for a direct to mass spec, it's quite impressive that most of the analytes fell within minus and plus 60%. And this is quite similar to sometimes liquid chromatography method. So we found that these values are, while there's high, but they're still impressive for a direct to mass spec technique. In terms of the effects of the matrix on the ion ratio of the qualitative, for identification purposes, the ion ratio between the quantitative ion and the qualitative ion, we found that 71% of the analytes fell within 10% ion ratio deviation when compared between the matrix and the reagent, and all of the analytes fell within plus or minus 50%, which is really quite impressive for a direct to mass spec technique. In terms of linearity, although we used only one internal standard per ionization mode for all of these analytes, we found that 55% of the analytes achieved R squared, which is better than 0.99%. And as you can see, like the other values, it's still very impressive linearity results despite the use of only one internal standard per ionization mode. In terms of accuracy and precision, also the majority of the analytes, when spiked at 0.4 and 0.75 and 1.5 times the MRL value or tolerances, it still fell within a quantitative range, which is 70 to 130% and less than 25% RSD. I'm going to move on to the next, quickly to the other technique of direct coupling of SPME to mass spec, which is direct analysis in real time, DART. As you can see here, it's very similar. It's almost the same approach like the one we used in case of LCMS, the same pins, but instead of the final desorption step, which was in desorption, here we desorb the pins using DART. And due to the nature of DART, we could only analyze 53 veterinary drugs. And in terms of results, here's a quick result for the accuracy and precision. Also, we find it like impressive for a direct to mass spec technique that the vast majority of analytes, when spiked at 0.5 and 1 and 2 times the MRL values, fell within a quantitative range, which is 70 to 120% accuracy and less than 25% RSD. Last, I'm going to talk quickly about untargeted metabolomics using SPME and liquid chromatography and mass spectrometry using an Orbitrap here. I'm going to be talking briefly here. Most of the results that I presented before are already published and the papers are mentioned in the slides. I'm going to be talking very briefly about the metabolomics because we are still working on the manuscript. Briefly, what we did here, we compared 100% beef and 100% pork and different percentages of beef and pork, and we analyzed them using SPME, LCM, SMS, using the same technique that the protocol that was used for the LCMS approach, and we found that we can easily distinguish between pork and beef, and we can also, although we cannot distinguish 1%, but we can distinguish 5% pork. So if the beef has been mixed with 5% pork, we were able to tell using this technique. Also using this technique, we were also able to identify the features that caused this separation between pork and beef, and we were able to identify and confirm these features that were responsible for this separation. Finally, as a summary, as we saw with SPME, whether it's coupled with LC or direct to mass spec, we saw minimal matrix effects, and we can perform screening and quantitative studies. In case of direct to mass spec, we were able to achieve very rapid results, less than 1 minute analytical runs with minimal solvent use. Although the results I presented are only for beef and chicken, it can only be applied to various animal tissues and other food samples. In terms of future considerations, what we're looking for is to kind of synthesize a new coatings that are capable of extracting very polar analytes, because unfortunately HLB, although it still extracts polar analytes, but it's not very effective for very polar analytes. And also we're looking forward to couple these techniques with portable MS instrumentation for in vivo or on-site sampling, and hopefully this technique will help us do analysis on the spot in supermarkets or even before slaughtering the animals. And finally, like this work, of course, has been done with the help of all of the team at Dr. Pavlishin's group. I'd like to thank all of them. I have the names here. I'm not going to go over them. Thank you, everyone, for attending this presentation, and I'm open for questions. Thank you very much.